Animal cell model number 1. This is a generalized animal cell. We're going to look at several different structures and their functions. The first structure I want to look at is the nucleus. The nucleus contains your DNA in the form of chromatin, and you can see the little chromatin strands in there. And the nucleus is surrounded by a nuclear membrane with nuclear pores. And inside the nucleus, we have the nucleolus. The nucleolus is a very specialized section in the nucleus that produces ribosomes. If we zoom in and look a little closer at this part of the animal cell, we can see the chromatin strands of DNA here in the nucleus. And in this part of the nucleus is where messenger RNA strands are generated through a process called transcription. The messenger RNA then leaves the nucleus through these nuclear pores, comes out into the generalized cell, into the cytosol or cytoplasm, where they interact with ribosomes to generate proteins. Here is the nucleolus. The nucleolus manufactures ribosomes. These structures are called mitochondria. Mitochondria produce ATP through a system called cellular respiration. And mitochondria are a unique organelle in that they have their own DNA, which is unlike the DNA of the host. This gives rise to the hypothesis that at some distant past, bacteria formed a symbiosis with primitive cells, and this is how mitochondria got included in today's modern cells. Now we will take a look at the endomembrane system. The first part of this system is called the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is often abbreviated SER. It contains enzymes, produces and digests fats and membrane proteins. Next is the rough endoplasmic reticulum, often referred to as rough ER. It gets its name because there are numerous ribosomes attached to it. The rough ER transfers materials through the cell and produces proteins. There are two types of ribosomes in the cell. There are the bound ribosomes, which are attached to the rough ER, and there are free ribosomes, which float around in the cytosol. We can take a closer look at these ribosomes. We can see the bound ribosomes attached here to the rough ER, and we can see some free ribosomes floating around here in the cytosol. The bound ribosomes make proteins that are exported or included in the cell membrane, certain transport proteins or gate proteins in the plasma membrane, while the free ribosomes make proteins that function within the cytosol of the cell. This layer is called the basal layer, and its function is determined by what type of cells it is associated with. Then we have collagen fibers. Collagen fibers provide strength and structure in underlying connective tissue. And this is a fibroblast. Fibroblasts function to build fibers. These white structures are called peroxisomes, which are formed in the Golgi apparatus. They contain oxidative enzymes and are used to rid the body of toxic substances. Then we have the yellow structures, which are lysosomes. Lysosomes contain digestive enzymes to either break down anything that is ingested by the cell or worn out organelles that accumulates in the cell. Then we have the Golgi apparatus. The Golgi apparatus is sort of a shipping receiving facility. The Golgi apparatus has many functions, but a couple of the functions are processing and packaging proteins. They receive them on one side of the Golgi apparatus called the cis or CIS face. They encapsulate them, turn them into a vesicle, which comes out the trans face. These Golgi bodies also transport lipids. These structures are secretory vesicles that have broken off of the trans face of the Golgi apparatus and are generally usually filled with a protein or a lipid that's being moved either around the cell someplace else or is about to be exocytosed out of the cell. This structure is the centriole. The centriole plays a very important role in cell division. This part of the cell that we're pointing at here is the cytosol or cytoplasm, and we can see the microskeletal fibers running through the cytoplasm. And here at the top of the cell, we see little microvilli. Microvilli basically are used to increase the surface area of a plasma membrane. This gives more area for diffusion to move back and forth across a plasma membrane. Here we see some desmosomes. Desmosomes are connections between adjacent cells, and we can see these little fibers, and there would be fibers on the other side here, and it forms a little connection holding cells together.